A few days ago, many people suddenly saw a scary word appear again in the headlines: hantavirus. And the reason sounded a little too familiar: a cruise ship outbreak. Honestly, after the whole world went through with COVID-19, seeing words like virus, cruise ship, and quarantine together can immediately make people nervous. Some people even started asking, are we doing this again? So today, let's walk through this story together. What actually happened? What exactly is hantavirus? And most importantly, why do infectious disease experts believe this situation is very different from COVID-19? The story started in May 2026 on a Dutch expedition cruise ship called MV Hondius. The ship had departed from Ushuaia, Argentina on April 1st for a South Atlantic expedition voyage. The route itself looked amazing: South Georgia Island, St. Helena, Cape Verde, and later the Canary Islands in Spain. Basically the kind of trip where passengers probably planned to come home with beautiful penguin pictures and nature documentaries on their phones, not medical quarantine stories. The ship carried around 147 passengers and crew members from at least 12 countries. But during the trip, several passengers developed severe respiratory symptoms. As the situation evolved, by May 13th, 11 people were considered suspected or confirmed hantavirus cases, and sadly, 3 passengers died. The virus involved was identified as the Andes strain of hantavirus. That detail is important because among all hantaviruses, Andes virus is one of the very few known to occasionally spread from person to person after prolonged close contact. Eventually, the ship was allowed to dock in the Canary Islands. Passengers were then gradually sent back to their home countries under different monitoring and quarantine plans. Some American passengers were transferred to Nebraska for observation and medical evaluation. The CDC also issued alerts to doctors in the United States about possible imported cases. Now let's refresh our memory with a similar yet very different outbreak during the early days of COVID-19. Back in early 2020, the Diamond Princess became one of the world's first major COVID outbreak stories after docking in Yokohama, Japan. Around 3,700 passengers and crew members were on board. More than 700 people eventually tested positive, and around 14 passengers later died. At that time, scientists still knew very little about the virus. Vaccines did not exist yet. The outbreak became one of the first major warnings about how quickly COVID-19 could spread in crowded environments. So when another cruise ship outbreak suddenly appeared in the news in 2026, people naturally became worried. But when infectious disease experts actually looked carefully at this outbreak, their conclusions were surprisingly consistent. WHO Director General Tedros Adhanom Ghebreyesus stated that there was no sign of widespread community transmission. Acting CDC Director Jay Bhattacharya directly emphasized that this was not the new COVID. Dr. Michael Osterholm from the University of Minnesota explained that hantaviruses spread far less efficiently between humans. Dr. William Schaffner from Vanderbilt University also noted that Andes hantavirus usually requires very close and prolonged contact for transmission, making a COVID-like global pandemic extremely unlikely. So why are scientists so confident about this? Well, first, hantavirus is very different from coronavirus at the biological level. Hantavirus is not just one virus. It is a whole family of viruses called orthohantavirus. The name comes from the Hantan River near the border between North and South Korea. During the Korean War in the 1950s, thousands of United Nations soldiers developed a mysterious fever with bleeding problems and kidney failure. Years later, scientists finally discovered the responsible virus in a small rodent called the striped field mouse. Basically, a tiny brown mouse with a dark racing stripe running down its back, living near the Hantan River. That discovery also revealed one very important fact: rodents are the natural hosts of hantaviruses. Today, scientists have identified more than 50 hantavirus species worldwide, although only around 20 are known to infect humans. Scientists often divide hantaviruses into Old World and New World hantaviruses. Old World hantaviruses are mainly found in Asia and Europe, including Hantavirus, Sol virus, Pumala virus, and virus. These viruses usually cause hemorrhagic fever with renal syndrome, meaning they mainly damage blood vessels and kidneys. New World hantaviruses are mainly found in North and South America, including sin nombre virus and Andes virus. These viruses are more associated with hantavirus pulmonary syndrome, a severe lung disease that can rapidly lead to respiratory failure. One famous outbreak happened in the Four Corners region of the United States in 1993, the area where Arizona, New Mexico, Colorado, and Utah meet at one point in the southwestern United States. Several previously healthy young adults there suddenly developed severe respiratory failure caused by sin nombre virus, bringing major public attention to hantavirus in America for the first time. But the most important thing to understand is this. Hantavirus mainly spreads from rodents to humans, not from humans to humans. Usually, people get infected after breathing in tiny particles from rodent urine, saliva, or droppings. For example, cleaning an old barn, abandoned cabin, basement, or storage room full of mouse droppings without proper protection can create risk. Human-to-human transmission is extremely rare. In fact, among all known hantaviruses, only Andes virus has shown convincing evidence of limited person-to-person spread. And even then, transmission usually requires prolonged close contact. Incubation time of hantavirus is very long, usually 2 to 4 weeks, making outbreaks spread far less explosive. This is already very different from COVID-19. COVID spreads between people extremely efficiently. Its incubation time very short, only 2 to 5 days. And because it infects the nose and upper airway very early, people can spread it simply by breathing, talking, coughing, or sneezing, even when symptoms are mild or before symptoms fully develop. Hantavirus behaves very differently. It tends to infect deeper tissues, especially blood vessel cells in the lungs or kidneys. Patients often become seriously ill quite rapidly, but because the virus is not heavily concentrated in the nose and throat, it spreads very poorly through casual daily human contact. And if you like molecular biology, the difference becomes even more interesting. COVID-19 has those famous spike proteins, basically little grappling hooks, that help the virus tightly attach to human cells. Hantavirus does not have these spike proteins. Instead, it uses less efficient surface glycoproteins to attach to cells. COVID-19 mainly binds to ACE2 receptors, which are widely distributed throughout the body, including nose, throat, lungs, intestines, and many other tissues. This gives COVID many opportunities to infect cells and spread rapidly. Hantavirus mainly binds to molecules called integrins, especially B3 integrins, which are found on a much narrower group of cells, especially blood vessel cells. In simple terms, COVID has access to many doors throughout the body, while hantavirus only has access to a few specialized entrances. Another important difference is actually written directly into the viral genome itself. COVID-19 is a positive sense RNA virus. In very simple terms, its genetic material already works almost like ready-to-use mRNA. Once the virus enters a human cell, the cell can quickly read those instructions and immediately start making viral proteins and new viruses. Hantavirus is different. It is a negative sense RNA virus, meaning its genetic material cannot be directly read by the host cell. Before the virus can produce proteins, it must first generate a positive sense RNA copy. Only after this extra step can the cell begin manufacturing viral proteins and assembling new viruses. Of course, some negative sense RNA viruses, such as influenza, have caused pandemics. However, influenza has special mechanisms that make it far more transmissible, and we will not discuss those here today. Diagnosing hantavirus infection can be tricky because the early symptoms are very nonspecific. Fever, headache, muscle pain, nausea, cough. Honestly, early hantavirus can initially look like flu, COVID, or many other viral infections. That is why laboratory testing is extremely important. Doctors often look for low platelet counts, elevated white blood cells, kidney injury, or signs of fluid leakage in routine blood tests. Antibody testing, especially IgM antibodies, is commonly used for diagnosis. PCR tests can detect viral genetic material directly. In some advanced laboratories, sequencing technologies can even identify the exact hantavirus species involved. Chest CT scans may show rapid fluid accumulation in the lungs in severe pulmonary cases. Currently, there is no universally effective antiviral drug specifically designed for hantavirus infection. Treatment mainly focuses on supportive care and early intensive medical management. Patients with severe lung disease may require oxygen therapy, ventilators, or even ECMO support. Patients with kidney-related disease may sometimes require dialysis. Some studies have explored antiviral drugs like ribavirin, which may provide partial benefit in certain old-world hantavirus infections, but supportive intensive care remains the most important treatment. Fortunately, prevention is relatively straightforward. The key is avoiding contact with rodents and their waste. People should avoid breathing dust from rodent urine or droppings, especially while cleaning barns, sheds, cabins, basements, or abandoned buildings. Health authorities recommend ventilating these spaces first and using disinfectants instead of sweeping dry droppings into the air. Controlling rodent infestations and storing food properly are also important preventive measures. For patients infected with hantavirus, especially the Andes strain, family members should avoid sharing towels, utensils, or toothbrushes, and should minimize very close, prolonged contact until the patient has recovered. And unlike COVID-19, there is generally no need for widespread lockdowns, panic buying, or fighting strangers online over masks because human-to-human spread is extremely uncommon. We are heading to the end of this story. Thank you for watching. See you soon.